Hello everyone. Today, we're going to discuss an intriguing topic, microbial identification techniques. As we know, there is a vast array of microbes including bacteria, fungi, protozoa, viruses, and archaea. These microbes play crucial roles in the natural world and human life. However, since microbes are typically invisible to the naked eye, scientists have made various attempts to identify and utilize them by developing a range of methods and techniques to examine their morphological, physiological, and genetic traits and differentiate microbial species in samples. Microbial identification methods can be classified into traditional and modern techniques. Traditional methods include morphological observation, biochemical tests, and physiological tests. Modern methods encompass gene sequencing, PCR, and MALDI-TOF. Today, let's explore one of the oldest techniques, the gram staining method, invented and named by Danish botanist and pharmacology teacher Hans Christian Gram in 1884. Although the gram staining method cannot provide detailed information about bacteria, it remains an essential tool for bacterial identification and classification. It is particularly valuable in clinical laboratories for quickly determining the cause of infection and in choosing appropriate antibiotic treatment plans. The basic principle of the gram staining method is to classify bacteria into two major categories, gram-positive and gram-negative, based on the structure and chemical properties of their cell walls. In the cell wall, which forms a layer around the majority of bacterial membranes, peptidoglycan, a compound containing sugars and amino acids, can be found. Gram-positive bacteria have a cell wall primarily composed of a thick peptidoglycan layer, whereas gram-negative bacteria have a cell wall consisting of a thin peptidoglycan layer and an outer membrane. As a result, gram-positive bacteria can absorb crystal violet dye and turn a dark bluish-purple color, while gram-negative bacteria can easily have the crystal violet dye washed away when soaked in alcohol and then appear pink under the influence of safranin dye. Gram staining provides two important characteristics, the gram reaction or cell color, and cell morphology, the shape of the cell. Bacteria cells can be spherical, rod-shaped, or spiral, with nearly all clinically significant bacteria falling into one of these three categories. As an ancient method, the gram staining technique has not faded from history. Instead, it continues to be a widely used technique applied in the following areas: bacterial classification, bacterial detection, bacteria morphology research, bacteria culture control. So how do you perform a gram stain? First, use a wax pencil to draw a circle on a microscope slide. Next, add a sterile saline or deionized water to the center of the wax circle. Then use a sterile loop to touch the edge of an isolated bacterial colony on an agar plate. Place the loop into the drop on the slide and rotate it evenly to distribute the cells, creating a thin layer. It's important to use cells from a single colony, as this could represent only one cell type and not a mixture. Ideally, the culture should be 18 to 24 hours old, as older cells may show variable results. Alternatively, you can use a swab from a clinical or environmental sample. After at least 10 minutes, when the liquid has completely dried, quickly pass the slide through a flame to fix the bacteria onto the slide. Next, add a few drops of crystal violet dye to the bacteria until the wax circle is filled with the dye. Let it sit for 1 minute. Then, dip the slide in a beaker of water 2 to 3 times to wash off most of the dye. Add a few drops of grams iodine to cover the bacteria and let it sit for 1 minute. Rinse the slide in a beaker of water until most of the iodine is gone. You can use a second beaker of water to ensure most of the iodine has been rinsed off. Now, add a few drops of decolorizer. Allow the 95% ethanol decolorizer to flow over the bacteria at a 45 degrees angle until the flow becomes clear. Then, rinse the slide with water. Add safranin dye as a counterstain, covering the bacteria, and let it sit for 1 minute. After 1 minute, thoroughly rinse the slide with water. Be careful not to overwash the slide and let it air dry. You can tilt it on a paper towel or sink to dry, or gently blot it with a lint-free absorbent paper, but do not wipe the slide as this may remove the cells. Now, you can examine the slide under a microscope. First, focus the image using a high-power dry objective at 40x, then without moving the slide, switch to a high-power oil immersion objective 100x for observation, resulting in a total magnification of 1000x. On the slide, you'll be able to see two types of bacteria: purple gram-positive cocci and pink gram-negative rods. Yes, it's that simple. However, gram staining can only be used for preliminary identification of bacteria and cannot determine the specific species. Nonetheless, it's sufficient for guiding the rapid selection of antimicrobial agents in clinical settings. That's it for today's sharing. Goodbye.